Hello, Professor Clark here, and we're going to talk about bacteria. So within this chapter, we'll discuss the prokaryotes, some of the variety of bacteria that we may encounter in and around eukaryotic living organisms, and also something that I call the extreme sport bacteria, the archaea. So let's get started. I'm going to go to our PowerPoint. This is found from chapter 15 in your textbook. And we're not going to utilize all of chapter 15, just some of it, just the material that pertains to the prokaryotes. So here we go. Bacteria, first of all, they're everywhere. Pro means before, karyo is the nucleus, referred to the kernel of the nucleus. So prokaryotes evolved before eukaryotes, and they also were discovered after scientists had discovered eukaryotic cells, those with a nucleus, because the prokaryotes are really small. We're going to be able to see them in our lab using the 100x oil immersion lens for those of us doing labs on campus. But anyway, so this is the term that we have, prokaryote. They're everywhere. The number of prokaryotes, a single handful of soil greater than the number of people who ever lived. I don't know how anyone can even know that. By the way, most of the bacteria in the soil is called Bacillus subtilis, and that's one of the specimens we may see in lab this week. E. coli found on the surface of your intestine. So here it's been given a fluorescent screen, it's called a fish screening, and so they seem to glow. And these are rod-shaped bacteria. We see some bacteria that live outside of a host cell. They're somewhat parasitic. They're deriving nutrients from that host, and it's not to the advantage of the host. So all these extracellular. Some live inside a host cell. Here's one bacterium that's actually inside a host cell. This is certainly not to the advantage of the host. And so the bacteria is considered a parasite. But oftentimes bacteria is living in or on a surface of the cell, and it's not pathogenic, it's not parasitic. Just like those enteric E. coli bacteria, they actually offer a significant benefit both to the digestive tract and to hormone production for the living organism. Some bacteria live in the gut and tubes of animals and help them break down that polysaccharide cellulose. So the methane gas that cows release on a regular basis is the byproduct from the fermentation of the cellulose. So the branch polysaccharide gets into the gut tubes of cows and rabbits and rats and koalas, and then the bacteria breaks down that cell wall of the leaves and the grass and the things for them. So otherwise, it would be an unedible source of food to the cow unless it had that bacteria. So many animals have an extra pouch or cecum that's filled with these bacteria. For us, it's our cecum. In something like a carnivore, the cecum is quite small. They don't really break down and digest cellulose. In something like this koala, the cecum is long and coiled, extended surface area, because they're ingesting really nothing else except the leaves of this eucalyptus, and they are relying on the bacteria to ferment or break down the cellulose. Here's the pointy part of a pin, the tiny pointy part, and we see bacteria here covered. And some of the terms you might see for bacteria, I might say the term prokaryote, which means cell lacking a nucleus. Remember, prokaryotes are definitely alive. They definitely have DNA, they have ribosomes, they make proteins, they do all these living characteristics, they just don't keep their DNA in a nucleus. That's what prokaryote means. Bacteria are one of the groups of prokaryotes. Microbe just means it's really small, and we need a microscope to see it. So microbe is probably not the best term, but you might see it anyway. So here's what we see. There's a YouTube video on here that I've linked. I want you to take a look at it on your own, and it's white blood cell, one of a human white blood cells tracking and going after bacteria. The white blood cell has detected the proteins in the plasma membrane on the surface of this foreign cell and are moving towards it, chemotaxis. They're moving towards this chemical signal in order to ingest it by phagocytosis and kill it. But anyway, here's some bacteria on the run from the white blood cell. I suggest you watch that. The functional and genetic adaptations to bacteria have contributed to their success. Their basic structure and cell design has contributed to their success. And remember, success in biology class doesn't mean that it has the most money or the biggest car or the most friends. It means, in fact, success means there's significant numbers of that type of organism, maybe in a variety of different habitats. Maybe they've manipulated one particular habitat en masse. So success means they can live and adapt and do very well in a particular environment, or maybe even in many environments. If we try to look at the DNA of bacteria, hopefully you've done that after taking Gen Bio 1, Gen Bio 2, molecular bio, cell bio, all of those courses we offer here at 4Cs. And then you can start to investigate some of the DNA. Because bacteria don't reproduce or share their DNA the way eukaryotes do, the way animals or even mammals or even vertebrates do. So they have an astonishing diversity of genetic material. These colorful plates show bacteria growing. If you take microbiology, you might do a lab that looks just like this. In the growth medium, we call that agar, A-G-A-R. In the agar, we can mix up various nutrients and find out what kind of bacteria grow best on sugar or on proteins or on milk sugars or on acidic conditions, vice versa. So you can mix different ingredients into the background material, put bacteria on it and see which ones grow. And then you can get their DNA, cell shape, and things like that after that. Most bacteria are one cell. Some will clump together and form a colony. When we pull our bacterial plates from the incubator, we see these colonies. That's where we want to scrape in order to take a look at some of the individual bacteria. And so you might find clusters of colonies. Sometimes bacteria will ooze this protective coating on the outside, called that a biofilm. I wake up every morning with biofilm on my teeth, and I anticipate you do as well. That's this material that we brush off in the morning. So the bacteria then have been building their biofilm on your teeth. Those bacteria also produce a byproduct, and that's what can eat through the enamel on your teeth and give you cavities. So we encounter bacterial biofilms in many environments. We encounter them on the roof or the lid of yogurt. So if you eat any kind of yogurt, once you pull the lid off and there's this film on the cover, that's actually the best part for you. That's where the living active cultures live. That's the bacteria that you want, acidophilus and things. We find that some bacteria are very helpful for your digestive tract, so you want those bacteria. So scrape that off the lid, mix it in, and eat it. This beaker, I imagine, is not something you want to ingest. I'm not really sure what's growing there, but it's gorgeous. If you leave some food in your refrigerator for a really long period of time, or maybe in the back of your car or somewhere, and you find a biofilm growing on it, may or may not have a particular odor to it, that's not to eat it. But you can think you created an incubation chamber that was adequate for some particular type of bacteria to grow and create its own biofilm. And here's that delicious biofilm that grows in your teeth. And sometimes it hardens over time and forms plaque. So when you go to the dentist, they have to scrape it off with a little scratchy pen and try to avoid those bacteria from then causing holes in your enamel. Generally speaking, bacteria, we can call them eukaryotes. Generally speaking, we call cells, pardon me. Generally speaking, we call cells either eukaryotic or prokaryotic. Remember, the eukaryotic cells have a nucleus and other membranous organelles. So eukaryotes include animals, plants, protists, and fungi. This little poster of green pond things, those are mostly the protists. Behind me, mitochondria is a membranous organelle, vesicles are membranous organelles, so is the chloroplast, so is the nucleus. Eukaryotic cells have this. Prokaryotes do not have membranous organelles. No nucleus, no mitochondria, no Golgi body, no ER, no chloroplast. Although prokaryotes have DNA, we can see it here, it's just loosely scattered throughout the cytoplasm. Sometimes it's in a region we call the nucleoid region, but don't worry about that. Prokaryotes can still make sugar. They can still make ATP. They can still make proteins. They can still move. They just don't require membranous organelles. Prokaryotes, as I mentioned, have DNA, but they don't keep it in the nucleus. And the DNA is often uncoiled or loose in an area we call a nucleoid region. I'm not going to ask you that. Occasionally, the DNA will form a little loop called a plasmid. I am going to ask you that. I think of a plasmid like a frisbee. And as soon as I see a frisbee, I'm going to toss it, and my dog's going to run after it and chase it. Bacteria toss their plasmids to each other. So one bacteria can drop or toss or send over a plasmid loop to another bacteria. Now, it isn't just a frisbee. It's not just a plastic toy. DNA holds the genes, and the genes are the instructions to make proteins. So if a bacterium tosses a plasmid loop to another bacterium, it's just sent over some of its genetic information. It's just sent the code or the recipe to make some type of structure. Animals do not reproduce like that at all. Animals do not share their DNA with each other. We take our DNA in an egg cell and a sperm cell, those two things fuse together and create an entirely new organism. It's a fusion of the genetic material. But the mom and the dad, they haven't combined their DNA with each other. They still have their own complete separate genome. Bacteria, however, do share their DNA with each other, and we call that those plasmid loops, or those small loops called plasmids. All right, more on bacterial reproduction in just a moment. Bacteria have ribosomes, so they create proteins, and they don't have those other membranous organelles I previously mentioned. They definitely have a plasma membrane because all cells on the planet have a plasma membrane. All the eukaryotes, all the prokaryotes, everything that's going to show up on a poster behind me. Everything has a plasma membrane made of phospholipids in a bilayer with proteins, cholesterol, and sugars. They're all made of the same thing. Additionally, bacteria have a cell wall. So they've got this plasma membrane, which in truth is very tough to see under our microscopes, but they also have a cell wall outside of it. One of the things that happens, remember the osmosis lab? Remember what happened when we put the sheep red blood cells into fresh water? Water goes towards the higher cellular concentration, water enters the cell, and in the hypotonic solution, the fresh water, the red blood cells explode. You can't kill bacteria just by tossing some fresh water on it. Water goes into the cell towards the higher cellular concentration, but the cell wall prevents rupture. So when you read in your textbook or hear me say, the cell wall helps the bacterium to maintain its cell shape and prevents it from bursting in fresh water, that's what we need. And think about what happened to the animal cell. Plant cells have a cell wall. It's built by different material. Bacteria have a cell wall, and it's constructed from separate molecules as well. So the plant cell wall and the bacterial cell wall have the same or similar function, but they're not made out of the same material. Plants are made out of cellulose. Bacteria, we're going to find the cell wall is made out of proteins and lipids. All right, so the cell wall is one of their most important features. Some bacteria also have little pili, little pelage, or furry appendages, hair-like appendages. And what the pili can do is they can help attach a bacterium to another bacterium, or they can hold a bacterium to a host. When and if you get strep throat, those bacteria enjoy the habitat of the mucous membranes of your pharynx. That's where they want to live. And they attach onto your pharynx. You somehow ingest or swallow or touch your dirty hands to your face, and it's carrying those bacteria. Those bacteria move through your oral cavity, find the mucous membranes of your pharynx, send out their little pili and hang on, and then start to reproduce. So these pili can have a number of advantages for the cell. Some pili actually trigger our immune system to crank up the temperature, meaning they're fever inducing. We call those pyrogens. Anyway, so the pili have a number of functions. As far as bacterial reproduction, so what happens here, it's similar to mitosis, not exactly the same, we're not dealing with centrioles. However, it's similar in that it's asexual. Mitosis is asexual. We don't have two parent cells that creates an offspring. In mitosis, we had one parent cell and it divided into two genetically identical daughter cells. Well, bacteria reproduce in a similar way. One bacterium will double up its genetic material, and then it separates or divides into two cells. We call this binary fission. The genetic material in each cell should be the same. This is not the plasmid loop story. This is how cells, bacterial cells, just increase their numbers. And it creates more bacteria that have the exact same genes. What I put on here is another YouTube caption that I think you should take a look at. And what this is showing is the rapid reproduction of bacteria. As long as they're given the proper food, maybe some moisture, the proper temperature, proper pH, bacteria can grow in a number of habitats. And what we often find, the bacteria that we study in our microbiology labs, is we study the bacteria that like to live on and in and around us, humans, because they're everywhere. They're on our hands, they're in our mouth, they're in our eyes, they're in our mucus. No matter how often we wash and scrub, we're covered with bacteria. And actually, that's a useful thing. If we scrub and scrape all the bacteria off our hands, we're going to be washing away some of the useful first line of defense bacteria that stave off or fight off some other more pathogenic things. It's an interesting scenario that we've developed, that we constantly are adding antibacterial and antimicrobial agents to our food, to our hands, to our clothing, to our shampoos. But we actually need the population of bacteria that coat our bodies, luckily go through binary fission and keep a lot of the more harmful pathogenic bacteria out. All right, so here's the plasmid story though. So in addition to binary fission, where one bacterium becomes 2, and then those become 4, and those become 16, and so on, with the exact same DNA, here's what can happen with the Frisbee game. Occasionally, bacteria can exchange their DNA, and they can do it where one cell directly transfers genes to another. That's what this picture is. So it's one of these extended pili, and to me, it always looks like the space station that's docked in with another space station. Now they have this little hamster tube for the astronauts to travel from one vehicle to another. But in this case, instead of astronauts, it's those plasmid loops. So imagine this, the bacteria on the bottom here, the one with the pili, it has the genes to make pili, to make those little extensions to maybe help it anchor onto a host cell. What if this bacterium locked in to another bacterium, and down this little hollow tube, it sends that plasmid loop with the genes to make pili. They break this connection. This cell, this bacterium incorporates that plasmid loop into its DNA, and before long, could be within 12 hours or 24 hours or 48, that cell is now making pili, and now it can attach it onto a host, and now it becomes pathogenic. This is one way that bacteria can transfer antibiotic resistance. The bacteria can transfer proteins, genes, sequences of information to each other, not intentionally that they're thinking about it. It's just one way that happens. It's not always useful, but in some cases, when they transfer those plasmids, it's then giving a whole population of bacteria that already exists. It's already, all of a sudden, giving them a new gene. If this interests you, you should take GenBio 1, GenBio 2, and we run these labs. We actually give bacteria genes to make them glow. It's called a P-Glow lab. They glow like a jellyfish, and then you can put the colonies of bacteria next to each other, and the bacteria will transfer that gene to the non-glowing bacteria. You give them 48 hours to grow, and you click on the ultraviolet light, and then all of a sudden, all the bacteria are glowing because they transferred this gene. Really fun. All right, there's another way. The second one is kind of like a vacuum cleaner, that one bacterium may just sort of vacuum clean up plasmids that they encounter in their environment. Maybe one bacterium has died or been broken apart or just released plasmids. I don't know why. But another bacterium in its environment can just move along and kind of like a vacuum cleaner, absorb plasmids from their environment and then take on that genetic material. And if you want to enter the world of microbial warfare, you might design some type of chemical or gene or molecule that kills off bacteria that are harmful. And you may leave those plasmids in the bacteria's environment, meaning you expose them to it. If they uptake that plasmid, you may be able to cause a self-destruct of that bacteria from the inside out. So there's all types of applications that we can manipulate, knowing how bacteria exchange their DNA. And there's another one using a viral vector. If you watch this YouTube program, you'll hear a little bit more about this transformation and absorbing of plasmids, and also how we can use viral vectors to put genetic material into bacteria that we may want to eliminate. But we'll just think of these two for now. When we plate up bacteria, it's possible to run labs where we kill them. That's really fun. So what's happened on this agar plate, the scientists have smeared out some bacteria, put it in an incubator, usually grows at our body temperature, 98 point something degrees, wait 1 to 2 days, and if we scraped off the smear of a colony, we could identify individual bacteria under the microscope. But another thing we can do is we can take, these are spores from a fungus. And so what's happened is that this fungal spore releases these enzymes called exoenzymes that it turns out kill the bacteria. Oh my goodness, we found an antibiotic. And one of the first antibiotics discovered and published and tested was, of course, from the common mold penicillium. We call that penicillin. So the environment is full of fungal spores. And so if you're running a bacterial lab, it's really important not to expose your bacterial cultures to any of these spores that may be occurring in the environment. Through the heating system, through the ducts, they come in on your clothes, they come in every time we open the door, etc. So here we have the zone of inhibition. And you can monitor how fast does it take this fungus, the enzymes in this fungus, to kill the bacteria. How fast, how far, how long? Does it clear the whole plate? If you drop this fungal antimicrobial component in the center, does it clear the whole plate of the bacteria? If you let it grow for 2 days, do the bacteria grow back? Are there some that have resistance and then they recolonize the plate? So here's another game in the world of microbial warfare. Not a game, not a game, but an experiment that we run in our lab. Not in Survey Bio, but as we move through some of the other classes, which are welcome to take, move along in that process. So we don't consider sex when we think about bacterial reproduction. It's asexual, it's that binary fission, but bacteria are able to transfer the genetics from one group of bacteria to another, including that antibiotic resistance. And you may know, if you've ever been given a prescription for antibiotics, the doctor may say, take the entire prescription, even if you feel better in 2 or 3 or 4 days, keep taking the entire prescription. You might think, no, I don't want to do that. I want to keep the other second half of this prescription in my refrigerator, so the next time I get strep throat or whatever it is, I don't have to waste time to go back to the doctor. But what you're doing is you're training the bacteria, maybe the ones that cause strep throat, to get stronger. The first couple of days of treatment, you might have killed off all the weaker bacteria, and the ones that may have some degree of resistance are kind of hanging out on the periphery, and your immune system cells start taking them out. But if you don't continue taking antibiotics, and your immune system cells then have to keep working on all the bacteria, the antibiotic resistance ones are going to come back in full force. And the next time you try to take that antibiotic, it's less effective because you've essentially trained the bacteria to become resistant because they played that ultimate frisbee game with the plasma groups. So take the whole prescription, even if you feel better. All right. A couple things with prokaryotes are the first form of life on the planet. That kind of sounds like a test question on the final for biology. What was the first life form on this planet? Bacteria. And we most likely think they evolved more than once. It's not like there was one bacterium, then there was 2, and then all of a sudden everything else. It's probably a gradual process. Well, it is a gradual process, but it probably occurred in more than one place where organic molecules coalesced, formed some type of cell-like structure, and it started linking amino acids together to make protein. But anyway, we think, the scientific community, that bacteria may have evolved in really extreme environments, and I'm going to talk about those extreme environments in just a minute. We find some fossils, not the bacteria, but the chemical trace of fossils in rocks that are literally 3.5 billion years old. There are areas on the planet where we have rocks that we can estimate the date, and they go back to the beginning of the formation of the Earth, and that is astounding to think about. So as soon as the Earth cooled and oceans formed, we think we had life on the planet. It happened pretty quickly. Prokaryotes get divided into 2 ginormous groups. We call those the domains. As we go through taxonomy, remember, did King Philip come over for good spaghetti? Domain, kingdom, phylum, class, family, order, genus, species. Domain, that's the top. These are the domains. We have the domain bacteria and the domain archaea. These are both containing cells that lack a nucleus. We're in the eukaryote domain. These are cells that have a nucleus. The animals, the plants, the protists, and the fungi. So there's some ancestral unicellular prokaryote that gave rise to what we call the archaea and the bacteria. All right, the archaea. This is not taken from your textbook. This is from the GenBio textbook. I don't think our book even has it in the chapters. But if you wanted to, you could compare the bacteria and the archaea, they're both prokaryotes, cells without a nucleus. You can compare them to each other, and you can compare them to the eukaryotes, the cells with a nucleus. I'm not going to ask you to memorize this list because I don't know the application of all of these factors. But I do know that the bacteria and the archaea have some similarities, like lacking a membrane-bound nucleus, lacking membranous organelles. They have these plasmid frisbee loops, the circular chromosomes that they can toss to each other to transfer their DNA. But then there's a number of things that are different. Some components of the lipids, some components of their response to chemicals, some in portions of their genetic code. And we don't need to memorize all that. The archaea, which we'll talk about in just a minute, I call these extreme sport bacteria, really old. Archaic makes me think old, like archaic. The archaea and the eukarya have some similarities, like lacking the sugar protein wall we'll find when we do our gram staining of bacteria. Some of these introns, of course, with genetic code and histones. So there's a link, there's some similarities between these three groups. So even as I go back to this slide, we think of the archaea and the bacteria as very similar in that they lack a nucleus, but there's the archaea that also have similarities to the eukarya. Everything on the planet can go into one of these three groups. These domains are enormous superseding groups just based on that very basic cellular structure. All right. For the regular old bacteria, these and the domain bacteria, they live anywhere around us, in us, on us, around us, in our soil, in the ocean, in the freshwater, in the puddle, in your blood, in sheep blood. In anywhere we find eukaryotes, we find regular old bacteria. And in lab, we're going to look at those and manipulate them. And those are the ones on this poster behind me. These are what we call the domain bacteria. This group, the domain archaea, I don't have any of these in our lab. And we can access them on the internet, but I don't have containers of archaea, like I do have containers of bacteria that we order from our supply house to run our microbiome labs. I think of the domain archaea as the extreme sport bacteria, and they live in areas of extremes, too extreme for any other living type of cell. Here we go. If something's called a thermophile, they love heat. Phile or philic means love it. Thermo in this temperature, in this setting means temperature. And we can have extremes of hot or cold. I only have a picture of an extreme hot. These are these geysers that we see in Western United States. In these puddles, if you scoop something, a piece of equipment in, and look at those bacteria under the microscope, you see, and these have had a fluorescent stain added to them. You see that there's bacteria. And the bacteria have also made this crusty edge. But there's no frogs in here, there's no fish, there's no seaweed, there's definitely no swimming. This is hot, and it's also toxic. But the bacteria seem to thrive in it. We see green grass not that far away, but this is enough for the bacteria. Extreme temperature, we call these the thermophiles. If we went out into California, these are salt evaporation ponds, and it's an area where salt is created, so that ocean salts are that people use for their food. And so we have these vast evaporation ponds. The weird colors are from the salt-loving bacteria. Halo means salt, halophile. So there's no birds lining on the edges, there's no fish in here, there's no human swimming. In these salt evaporation ponds, there's groups of these archaea, extreme spore bacteria, that enjoy and love this environment. This place might be 10 times saltier than the ocean, 30% salt. It could be like the Dead Sea, or people float. It takes excess amounts of weights to even get in there to go diving. I met some divers who were going into the Dead Sea to go diving for archaea halophile collection. And they had to weight themselves down to go even sink below the water to go swim. It seemed like a sort of a risky adventure to me. But these are the halophiles. Then under the microscope, they look at the bacillus shape. We'll learn the shape in just a moment. There's some bacteria that love it very acidic, acidophile. And so here's a sulfur spring. This is near Mexico, sort of Central America area where the limestone has collapsed away. And we see accumulations of hydrogen sulfide, and it's extremely acidic. Again, no fish, no crabs, no jellyfish, no. This is extremely acidic. And after you go through these caves, you have to wear a mask, not only to protect your lungs or the air you breathe in, but you also have to cover your skin. These little drops of goop have been created by these bacteria. And if they fall on you, they have a pH of about 3. We remember that, 2 or 3. And if you go and look into the bacteria, again, into these statites, as the scientists have called them, the microbiologists, we see bacteria. So these live in areas around these caves, near these hydrogen sulfide regions, where it's extremely acidic, no other like it. And then the last weird one that I'm going to talk about are these bacteria that live in and around the hydrothermal vents. If you sink to the bottom of the ocean out near the Galapagos Islands, we find 2 miles down, it's pitch black. This picture, the submarine had a headlamp that it turned on once they got to the bottom. But there's no sunlight in these environments. And we find these hydrothermal vents where chemicals from the ocean are spewing up and creating these upward molten cavities. And at these smoker chimneys, we find along the edges and even within the animals, these tube worms and other animals that live nearby, they're loaded with these bacteria, these type of archaea, rather, prokaryotes, that are able to use the chemicals of the earth to accomplish ATP production. That'll be another lecture. So these are some of the extreme environments where we find the archaea. And as scientists study them, they think, well, perhaps, maybe, this is where life got started. Not so much on the surface of the planet, in these primordial soup ponds, but maybe, if you've heard about that, but maybe in these extreme pockets, life got started driving these chemical reactions, and from there, we have the evolution of the rest of life forms on there. Just a theory. What we're going to need to do with bacteria, back to the regular old bacteria prokaryotes, we're going to think about where do they live, can they tolerate oxygen. We're going to do a gram stain in lab, either actually do it or look at it. We're going to want to identify bacteria by their shape and how they're arranged. So that's what we're going to do at this introductory level lecture to bacteria, prokaryotes in general. So we've already discussed habitats. The archaea are in this extremes environment, temperature, pH, salinity, lack of light. And the bacteria, regular domain bacteria, are anywhere in and around the eukaryotes, in the ocean, in the water, on our skin, in a cow, etc. As far as oxygen use, bacteria that use oxygen, we call them aerobic. It's a word that you're probably familiar with. We're certainly aerobic. Some bacteria use oxygen in their process of making energy. Some bacteria cannot tolerate it. And if you expose these bacteria to oxygen, you can destroy them. It's one of the ways that some materials and fruits like strawberries and things are clean. Instead of scrubbing them, they're irradiated or blasted with ozone linked form of oxygen. So some bacteria are actually anaerobic. They cannot tolerate oxygen. Then there's a facultative anaerobe. These are bacteria that prefer to live with oxygen, but you know what? They're fine without it. And that's very useful. And it gives us one of these insights as to how bacteria share their plasmid loops. So the facultative anaerobes could have been sort of a turning point. It could have been an offshoot of a branch. Got to take microbiology to study that a little bit more. What we're going to learn from lab is called the gram staining. This is named after an individual, Gram, so it has to be capitalized. And he's not the only microbiologist who wanted to do something with bacteria, put them into some kind of category. I sort of think of it when I'm walking through the supermarket and I'm looking at all of the salad dressings. Like, how do they organize these salad dressings? They can do, cream base versus oil base. I don't know, but there's all kinds of ways to organize things. Scientists love to organize. And so what we do with bacteria is we actually organize them based on their cell wall. because Johannes Graham found out this little bit of information. We can put bacteria into the purple group or the pink group. Basically, here's how it goes. Some bacteria, we find out, have a really thick cell wall. It's made of proteins and sugars, peptidoglycan. You can just say thick sugar protein, protein sugar cell wall. And what Graham discovered was that when you have these bacteria, if he covered them with this purple stain called crystal violet, the purple stain sucked into the sugar protein cell wall and made the bacteria look purple. So when we look under the microscope and we see purple stained bacteria, decided to call them Gram positive. But then Graham noticed that not all the bacteria will turn purple. And so he played around, experimented with his combinations, and actually it's not completely factual because there's a group of scientists that came up with a counter stain. But we'll just go ahead with the story. So the Gram negatives are bacteria that do not adhere this purple stain because their cell wall has some of these sugar proteins, but also has an extra lipid sugar layer, an extra slippery lipid layer. And so when we add this deep purple stain, it slips right off these bacteria and they never look purple. But we can try again, and they call that a counter stain. And so it was discovered this material called safranin red. If you add safranin red to these bacteria, that will lock into the slippery lipid, and then they look red. So when you look at this picture, you think, that's red. Well, it's sort of red, a pink, a light purple, however you want to describe it. So we have Gram positive purple, Gram negative are red. Keep going. So here's another picture of them. And we have the Gram positive bacteria. So we stain them. This thick sugar protein layer adheres in the crystal violet and they appear purple. The slippery lipid layer, the purple washes off. We go back and we try it again. We add another red stain to these and it does adhere. So the Gram negatives look red, reddish or pinkish. By the way, when you run this lab, you do the exact same procedure for all the bacteria. If you add the safranin red to the Gram positive, it doesn't matter because they're already dark purple. The red doesn't show up. So we do the exact same procedure for all the bacteria. And at the end of the lab, we have a handful of Gram positive and Gram negative. What does it mean? I don't know, just you can think about one way to categorize. We have oil-based versus cream-based salad dressing. It's one way to start thinking about what can I do with all this massive diversity of bacteria. So here's more information on it. Here's a little bit about Gram. So we get the sugar proteins turn purple and crystal violet. This one, there's this extra lipidy layer. The purple doesn't work. Try it again with red, and they look red, Gram negative. If you want to learn more about Gram and why he didn't come up with the perfect stain for the Gram negatives, but we still call it that, although he came up with Gram positive. All right, as far as shape of bacteria, this I'm going to talk about a little bit more during your lab. If you want to think about Gram positive versus Gram negative, some Gram positive, that's what you scrape off the lid of the yogurt and eat it. Acidophilus. And other ones cause Clostridium, which is a really common virulent urinary tract infection bacteria. Yikes! Some of them cause botulism, that flesh-eating skin disease and strep throat. Oh my goodness. But we can tend to be able to kill them off pretty well with a variety of antibiotics we've discovered. So which is worse, Gram positive or Gram negative? You can't come up with an answer because some of these Gram positives are really helpful for you. Gram negative. Here are some of the bacteria that live in your large intestine, and we want them there. They're enteric. They live happily inside us, and we want them there. We don't want them on our hands. We don't want them in our hamburger if we're eating that from a restaurant, and they end up in the upper portions of our digestive tract. But we do want them to break down cellulose for us in the rest of our digestive tract. Some Gram negatives don't bother us at all. They're photosynthetic. It's like this blue-green algae that we find on the edges of oceans and ponds. But some of them cause salmonella. That's what you get from licking raw chicken or licking turtle shells. Why would you do either of those? Pseudomonas is a common cause of pneumonia. Spirochete bacteria include everything from those that cause syphilis to Lyme disease. Goodness. Gonorrhea, meningitis, pneumonia. So a number of these gram-negative bacteria may be more problematic to our cells. But not E. coli when it's in its right place and not cyanobacteria. So we can't say one's better than the other. We can say, generally speaking, that the gram-negative bacteria may have some degree of antibiotic resistance. So when you go into the doctor for that quick strep throat culture and they send you back home with an antibiotic right away, they're kind of banking on the fact that that's probably what you have, the particular bacterium. And then sometimes you get a call back from the doctor and they say, you know what, we're actually going to change your antibiotic prescription. And you think, why? Well, it's just because they needed to wait for the bacteria to grow. It takes 24 to 48 hours. And then someone at the lab has done what we're going to do in lab. They take the bacteria that you had growing in your throat, for example, and then they grow them on a plate and they look them under the microscope and they see are they gram-positive, gram-negative. Right away, if they're gram-negative, they're probably going to change your antibiotic. If they're gram-positive, you probably have a general antibiotic that they prescribe pretty readily, unless you're, of course, allergic. All right, for the shapes of bacteria, we see your circle shape, your rod shape, and a spiral. So the circle shape we're going to call cocci is plural, coccus is one. This picture shows them going through binary fission. They're splitting. So it's not really the best picture of a sphere. This one's a little bit farther along. And then we see the 1 and the 2. So all these bacteria are breaking apart through that asexual reproduction method we call binary fission, and they're duplicating very quickly. The rod shape is called bacillus. Plural is bacilli. And this is also replicated by binary fission. So they can double up their DNA, increase their cytoplasm, split, and break. And now you got 2. So they can replicate very quickly as well. The spiral bacteria, the spirochetes, these are often pathogenic. It's like the one that causes Lyme disease. And spirochete bacteria, the spiral shape, often are problematic to human tissues and rapidly colonize and take over their host. Here's Borrelia burgdorferi. It's a type of bacterium. It may be carried by deer ticks, dog ticks. And when you bite, the center of the bite is probably somewhere around here. And the rings that people see with Lyme disease are the areas of the cells becoming necrotic. As that bacteria is replicating, it's increasing and increasing. But not everyone who gets Lyme disease has a bullseye. It's not always an indication. This indication, this bullseye is definitely a sign you need to be at the doctor's 2 days ago before the bullseye got that big. Get on antibiotics immediately. But not everyone that gets bitten by a tick carrying one of the Borrelia vectors will show that bullseye. All right, here's another kind of bacteria that causes Lyme disease. This is another Borrelia miyamotoi. I actually had this summer of 2019. Never saw a bullseye. This is not my blood. This is what my blood would have looked like if they had shared it with me at the lab. And there's that little spiral bacteria. Here's a white blood cell that just doesn't really know what to do about this. And here they are detected by PCR. So that was not fun at all, but it's fun. All right, so we go through the arrangements of bacteria. So the last thing I want you to know for the bacteria, and you either see them, or we can see them as a single, we can see them as doubles, we can see them in a chain that is strep, and we can see them in a spreading cluster that's staph. So now we're going to be able to mix and match it. We look at this bacteria, and it's purple, so it's gram-positive. And they're circles, so it's the coccus shape. And they're in chains, and the chain word is strep. So after examination and under the microscope, we can say, here's gram-positive streptococcus. And it's like we went to medical school and we didn't even know it. All right, then we'll keep going through those. We'll see a number of samples in lab, gram-positive, gram-negative, strep, staph. We'll see coccus shape, bacillus shape, and we'll talk about this bacteria a little bit more. All right, the last thing I want you to know about functions of bacteria, for sure, on the next exam, one of the essay options is going to be List and describe for me several useful functions for bacteria. Why don't we just want to kill them all? So these next few slides of maybe 10 or so useful functions, you pick 3 or 4 that stick in your mind that are interesting to you, because you're going to write an essay for me. So on exam 2, I'm going to ask, list and describe for me several, which means like 3 or 4 useful functions of bacteria. First one, bacteria break down dead stuff and they release the carbon. They break down dead plants and dead animals and dead mushrooms and other dead bacteria. So instead of all those trees that fall down or an animal that's hit on the side of the road, instead of just existing there and holding that carbon, bacteria break that organic molecule, those organic molecules apart and release the carbon. And maybe even release it as CO2 gas. And maybe then it's moved back to the ecosystem. Bacteria can take nitrogen gas and put it into a usable form for plants. Remember, nitrogen is a major component for proteins. We're made out of proteins. Most of our atmosphere is nitrogen gas. It's nitrogen triple bonded to another nitrogen. But our cells I can't break that bond. We can't get nitrogen to put into our amino acids just from breathing it in. We have to get our nitrogen from eating plants or eating some kind of food or animal that ate the plant. Nitrogen moves its way up the food chain. And bacteria take the nitrogen from the air and convert it into a form that plants can use. Thank you, bacteria, because without it, we wouldn't be alive. Bacteria form the base of the food web for many ecosystems. Any ecology class that you go to, remember you had like a food chain. Now it's more of a food web. And the base of that food web, whether it's a marine or a freshwater, wherever it may be, there's always bacteria at the base of it. You wipe out all the bacteria, that's the first step for food or nutrient transition, and then we wipe that out for the rest of the subsequent groups. Bacteria help things glow, and maybe you've seen Finding Nemo. And if you remember that some fish glow, and they may glow along their lateral line to, I don't know, flash a predator and distract them, or find food, or stun their prey, or all those reasons why animals want to glow. Fireflies do it to attract a mate. And oftentimes the luminescence, the glow factor in animals, is the result of some bacteria that they are kept inside pockets and nourish those bacteria, and as a result of for them. We use bacteria in the food industry. We take grapes and different plants and ferment them into wines and alcohols. We ferment cheeses. We use fungus for this as well, but we're not talking about fungi right now. That bacteria help in that whole fermentation process, and we have created all types of foods that we eat and drink. Bacteria in our bodies make vitamins for us. They make a vitamin B precursor, vitamin K. They link some of the fatty acids. So actually the building blocks that our bodies are made of are made by the bacteria living in your intestine, which is just such a bizarre thought. It's fantastic at the same time. Some bacteria are sources of food. They can be sold in health food stores. They're actually consumed as a source of food, like those bacteria in yogurt. We know that some bacteria can break apart the lipidy layers of hydrocarbons, like oil spills. And so in this area, bacteria are being sprayed on this oil spill to break apart those hydrocarbons, remember those non-polar fatty acid tails. And some bacteria can actually break those apart. We call this bioremediation. And you may want to investigate this more if you're taking an environmental science class, because then you have to ask, well, then what happens to the bacteria? And do they decompose themselves, or do they occupy another niche? It's always curious when we start adding one thing to the ecosystem that wasn't there before. Some bacteria help to break down human waste, and we see these little discs get covered with bacteria, and then they're tossed into sewage treatment plants to try and break down the material, so by the time the water is released back into the ecosystem, it doesn't have those same components to it, and some of that human sewage waste that's been broken apart can be used as fertilizer in pigs. This picture was years ago where one of the tanks that had all these little discs that had bacteria on it broke, And so these little discs were released, not to mention all the sewage as well, and that moved down the coast from New Hampshire all the way down to Cape Cod and beyond, but that was years ago. So if you ever find one of these discs on the beach, at this point, they're fine to pick up because they've been washing around the ocean for like 12 years, and there's no more bacteria or fecal matter on them anymore. But they did present quite a bit of a plastic trash hazard. All right, on your body, it's your first line of defense. So the bacteria that live on the edge of your nose and your mouth that are covering your body surface help to keep away other bacteria that may be more harmful, some fungi. So we call it our first line of defense. Oftentimes for female, if the female takes antibiotic to kill off some type of bacterial infection, it also inadvertently kills off the bacteria in the vulva, and then a yeast infection ensues. And a yeast infection is a result of a fungus called Candida albicans, normally a resident in the vaginal canal, but the bacteria keep it at low numbers. Once an individual female takes antibiotics, it's possible for those bacteria to die down, and then the fungus really have nothing to keep them in check. So often strep throat may be followed by a yeast infection. All right, some antibiotics and antifungals are actually made out of bacteria. So some bacteria fight off other bacteria. We can figure that out. We can put them in a cage match together and fight them off if we don't want them. And we can also use some bacteria to fight off fungi. Every time we're exposed to bacteria, fingers crossed, we build our immune system. And so in exposure to more bacteria, we train our immune system to learn how to fight off microbes should they encounter our system again. That's the whole point of immunization, vaccines, etc., whether it's for a bacterium or virus, and on and on. But as typical immune systems are exposed to many bacteria, we build our immune system and it makes it stronger. There's all kinds of exceptions in there in between, just a general statement. All right, the last one is kombucha. Yeah, this is the last one. Maybe you've had some. You could buy this in the market. It's really expensive. Maybe you have a friend that makes it. I have a friend that makes it. And it starts with this pancake called a SCOBY. And the SCOBY has a mixture of bacteria and yeast, a fungi. And the SCOBY is then given water and other materials to ferment. And you end up with this very healthful, somewhat acidic material that is helpful to fight off some unhealthy bacteria when you consume it. So if you're curious about kombucha, ask around. Maybe somebody you know makes it, and maybe you can buy some in the market and try it. It's a little bit acidic, sometimes gives an upset stomach, but there you go. So that brings us to the end of bacteria, the bacteria lecture. I will be talking about this a little bit more, again, when we're in lab and we're either plating bacteria ourselves and looking at them, or we're going online and doing some reference. But either way, good luck.